A very good morning, students. So we have already read about the dihybrid cross. Now we are learning the another law given by Mendel, that is law of independent assortment, that is directly linked to dihybrid cross. So according to the independent assortment or Mendel, two characters combine to form a new hybrid, right? So what are they trying to say? Suppose seed shape and seed color, there are two characters here. So these two characters can form n number of or possible combinations that can recombine and form a separate trait, right? Their trait can segregate and recombine in possible combinations. So we have seed shape, right? R means round and wrinkled, seed color yellow and green. So they make these type of new generation or new combination. So these combination will separate and form many gametes, like they can form RR, RY, RY, capital R, capital Y, capital R, small y. So we get to four combinations, and these four combinations can give in the F2 generation the phenotypic ratio is 9 ratio 3 ratio 3 ratio 1. So they can make every possible combinations to show. So this is all about the dihybrid cross or law of independent assortment. Now we are reading about polygenic inheritance. Poly means many, genic means genes. So when three or more genes control the expression of a same character, what does it mean? It means that one character, like we are taking an example of skin color or height, they are characters. So one character is controlled by one character is controlled by three or more genes. Three or more genes. For example, we can take a height that is controlled by gene A, B, and C. So here we see the additive effect. So what does it mean? Like we are taking three genes, and if we see capital A, capital A, capital B, capital B, capital C, capital C, here the 6 dominant alleles are present. So 6 dominant alleles can make a person tall. So person here will be the tallest. And if we see capital A, small b, capital B, small b, capital C, and small c has only 3 dominant alleles, capital A, capital B, and capital C. So the height would be intermediate. And if we see here capital A, capital A, capital B, capital C, so there are no dominant alleles present here. So the individual will be the shortest or dwarf. So dominant character is directly proportional. Dominant is directly proportional to the character. So more the dominancy will show, so the more character will be developed or give their effect. So this type of genes gives additive effect. So dominancy depend on the character, or character depend on the dominancy. More dominant will be the character, more it will show its phenotypic characters. So now we are already read about the Mendel's law. So the failure of Mendel's law is very clear. So you just need to open up your NCERT and just read it out. And now I am moving forward towards the rediscovery of Mendel's law. So do you know that Mendel's work remained unnoticed for nearly 35 years? Around 90s, three scientists, De Vries, Correns, and von Schirmack, independently rediscovered the Mendel's experiment. Around the same time, these scientists were learning or rediscovered about the or studying meiosis. So they noticed something fascinating. They noticed that when the Mendel's giving a term known as factors or genes, they identify the term chromosome. So the chromosome behave exactly like the factors. So we can conclude that during meiosis, they discovered that chromosomes separate, and during fertilization, they reunite. Exactly the same thing happens with the genes of factors. So what they say, that chromosomal movement means during the meiosis there is an anaphase where the chromosomes segregate on opposite poles. So the chromosomal separation or aggregation during the meiosis works similar to the Mendel's gene. So this is a rediscovery by Mendel's law by these three scientists. Now we are moving towards the chromosomal theory of inheritance. This is given by Sutton and Boveri. Sutton and Boveri were given this theory, and what they said? They said information pass from one generation to next generation to via chromosomes. So according to this theory, listen carefully. According to this theory, genes are located on chromosomes. Genes are located on chromosomes, and chromosomes occurs in pairs. And during meiosis, homologous chromosomes separate. Each gamete receives one chromosome from each pair, and after fertilization, the chromosome number is restored. So therefore, chromosome carry heredity information from parents to offsprings. That's why chromosomes are called vehicles of inheritance. Think of a school bus. Students are genes. If we are taking an example of a school bus, students are genes. The bus is the chromosome. Bus is the chromosomes. And without the bus, student cannot reach the school. Without the bus, student cannot reach the school. Similarly, genes travel through chromosomes. Genes travel through chromosome. Bus is the chromosome. So genes travel through the chromosomes. So what does it mean? That information passes from one generation to next generation via chromosomes. So this theory was proved by T.H. Morgan, Thomas Hunt Morgan. He is also known as flyman of genetics. Fly word comes from fruit fly or Drosophila melanogaster. So according to him, he proved the experiment. He proved the chromosomal theory of inheritance. How does he prove? He worked on Drosophila or fruit fly to prove his experiment. So why Drosophila? Because there are few reasons for choosing the Drosophila. So the first reason is its life cycle. Its life cycle is about 2 weeks. 2 weeks means 14 to 15 days around. So many generations can be studied very quickly. Second would be it produces hundreds of offsprings, hundreds of progeny, n number of progeny they have produced. So the large sample size means more reliable results. Third will be it grow easily on simple culture media. So they get easily grown on a simple culture media. And fourth is sexual dimorphism. Do you know the word about sexual dimorphism? Sexual dimorphism refers to the differences in appearance, anatomy, and behavior between male and female fruit flies. So the male, we can differentiate easily, we can distinguish easily between male and female. So male is large, male is small, and female is large. Male have solid tip on abdomen. So they have solid tip on abdomen, males. And in females, there are strips on the abdomen. So we can easily identify them. So this term is known as sexual dimorphism. Fifth is contrasting characters easily seen with low power microscope.